Can DNA be extracted from a mummy?
Yes, DNA can be, and has been, extracted from a mummy. However, the problem with extraction of ancient DNA lies within contamination from modern DNA. In order to minimize contamination, researchers usually try to get DNA from inside teeth or bone. Ancient DNA is being used to study the genealogy of the pharaohs of Egypt.
Why do animals defend territories?
Animals defend territories to protect their assets such as food, water, or mates. Animals will also defend the area where their offspring live. Because in evolutionary terms offspring are the ultimate asset.
How is a gene library made?
To create a gene library, scientists extract the DNA of a specific organism and use restriction endonucleases to cut it. The scientists then insert the resulting fragments into vectors and make multiple copies of the fragments. The number of clones needed for a genomic library depends on the size of the genome and the size of the DNA fragments. Specific clones in the library are located using a DNA probe.
What is nuclear magnetic resonance imaging?
Magnetic resonance imaging, MRI, sometimes called nuclear magnetic resonance imaging. NMR, is a non-invasive, non-ionizing diagnostic technique. It is useful in detecting small tumors, blocked blood vessels, or damaged vertebral discs. Because it does not involve the use of radiation, it can often be used where X-rays are dangerous. Large magnets beam energy through the body, causing hydrogen atoms in the body to resonate. This produces energy in the form of tiny electrical signals. A computer detects these signals, which vary in different parts of the body and according to whether an organ is healthy or not. The variation enables a picture to be produced on a screen and interpreted by a medical specialist. What distinguishes MRI from computerized X-ray scanners is that most X-ray studies cannot distinguish between a living body and a cadaver, while MRI sees the difference between life and death in great detail. More specifically, it can discriminate between healthy and diseased tissues with more sensitivity than conventional radiographic instruments like X-rays or CAT scans. CAT, computerized axial tomography. Scanners have been around since 1973 and are actually glorified X-ray machines. They offer three-dimensional viewing but are limited because the object imaged must remain still.
What happens in a cell if mitochondria are defective?
Since mitochondria are the energy producers of a cell, if a cell's mitochondria are defective, any cells that have a high metabolic rate will also be affected. There are metabolic poisons that affect specific aspects of mitochondrial function. These include cyanide, dinitrophenol, an ingredient of early diet drugs, valinomycin, and gramicidin. If mitochondria are damaged, they begin to leak free radicals that can alter DNA. Because mitochondria have their own DNA, it is also possible for them to be altered by genetic mutations. Mutations in mitochondrial genes are thought to play a role in degenerative neurological diseases such as Parkinson's and Alzheimer's.
Where does the carbon dioxide that we exhale come from?
The carbon dioxide we exhale is a result of the breakdown of glucose. During the energy harvesting phase of aerobic cell respiration. During this process, all of the carbon atoms, from the C6H12O6, are released as carbon dioxide molecules. Of the 6 carbon dioxide molecules generated, 4 are released via the Krebs cycle.
What is terminator gene technology?
This is a method of biotechnology in which crops that are bioengineered for a specific desirable trait, such as drought resistance, would contain a lethal gene that would cause any seeds produced by the plant to be non-viable. The lethal gene could be activated by spraying with a solution sold by the same company that originally marketed the bioengineered plant. Thus, the plants would still provide seeds with nutritional value. But these seeds could not be used to produce new plants. This technique would allow companies to control the product's genes, so that they would not spread into the general plant population. Seeds would have to be purchased by the grower for each season.
What is the function of the nucleolus?
The nucleolus is a large, spherical structure present in the nucleus of a eukaryotic cell. It is the site where ribosome subunits are assembled, and where both ribosomal RNA synthesis and processing occur.
What is the name of the early Jurassic mammal that is now extinct?
The fossil site of the mammal Hadrocodium wui was in Yunnan Province, China. This newly described mammal is at least 195 million years old. The estimated weight of the whole mammal is about 0.07 oz. 2g. Its tiny skull was smaller than a human thumbnail.
How does meiosis relate to Mendel's laws?
Mendel's law of segregation directly refers to meiosis. He had suggested that gametes contain characters that control specific traits. And these characters separate when gametes form. Gametes are formed by meiosis, in which the chromosome number is reduced by 1/2, from diploid to haploid.
In what way can forest fires be good for the environment?
Wildfires are critical to maintaining the integrity of forest and grassland ecosystems. Forest and grass fires, usually started by lightning, act as an ecologically renewing force by creating necessary conditions for plant germination and continued healthy growth to occur. The primary goal of fire management is to simulate the revitalizing aspects of natural fire cycles. Fire management also attempts to prevent large catastrophic wildfires from occurring by removing accumulated debris from forests. Seen throughout the American West every summer, these extremely intense fires are caused primarily by decades of fire suppression, which has allowed heavy fuels accumulated debris to build up. Ironically, by attempting to prevent natural fires, humans have only increased their prevalence.
Where do cancer cells come from?
When the reproductive rate of cells exceeds their death rate, the tissue becomes enlarged, forming a tumor. Although these cells are initially identical to the others in the tissue, they gradually take on characteristics of malignancy. The cancer cells reproduce rapidly and tend to be abnormally large or small. Malignant tumors grow 78 very quickly and invade other tissues. Cancer types are named for the location of the tissue that gives rise to the tumor and the organs involved. Genetics, viruses, or even environmental exposure to substances like those in cigarette smoke may cause tumor formation. However, not all tumors are malignant. Tumors that grow within a well-defined capsule are benign and unlikely to be life-threatening.
What mushroom has been identified as a fossil?
Mushrooms are rarely found in fossil records since there is little in the structure of a mushroom that can be fossilized. In 1992, researchers in the Dominican Republic discovered a fossil of a fleshy, gilled mushroom. The mushroom, Copernites dominicana, is believed to be 35 million to 40 million years old and is the only known fossil mushroom from the tropics. Microscopic examination suggests it is related to modern-day inky cap mushrooms.
What predicts whether one animal will help another?
Usually animals that help one another are of the same species. conspecific, and are most likely to be genetically related. The closer the relationship, the more likely helping will occur. This is demonstrated among vampire bats, where bats returning to the roost may share blood with those who have not fed. Most sharing occurs between close relatives, e.g., mother and offspring. However, unrelated but closely associated individuals may exhibit reciprocal altruism, where help is given to those individuals most likely to return the favor.
Why are cells so small?
Cells come in a variety of sizes and shapes. Bacteria are among the smallest, 0.2 to 0.3 µm in diameter, while cells of plants and animals are generally larger, 10 to 50 µm in diameter. Cellular size is determined by the surface/volume ratio required to maintain access to the substances and enzymes that cells need to complete their functions. For further understanding of the surface/volume ratio, imagine that a cell is the shape of a cube. The volume of a cube increases as does the length or diameter, but the surface area only increases as the cube does. Once the upper limits of surface area/volume ratio are reached, the cell generates more cytoplasmic volume than can be met by the small increases in membrane surface area.
What is the NIMBY syndrome?
NIMBY is the acronym for Not in My Backyard. It refers to major community resistance to construction of new incinerators, landfills, prisons, roads, and so forth. NIMBY is not in my front yard.
How did Hugo de Vries demonstrate sympatric speciation?
Hugo de Vries, 1848 to 1935, discovered a way in which a population could become a separate species while still sharing the same environment with other members of the species. The process, known as sympatric speciation, occurs almost exclusively in plants rather than in animals and involves a series of rare genetic accidents that can occur during the formation of gametes, eggs and sperm. As a result, gametes are formed as polyploids, that is, they have extra copies of each chromosome and thus are unable to match their chromosomes to others of the same species. Since these polyploids are forced to mate only with other polyploids in the population, they are reproductively isolated and considered a new species.
What is scientific notation?
Scientific notation allows scientists to manipulate very large or very small numbers. It is based on the fact that all numbers can be expressed as the product of two numbers. one of which is the power of the number 10, written as the small superscript next to the number 10 and called the exponent. Positive exponents indicate how many times the number must be multiplied by 10. While negative exponents indicate how many times a number must be divided by 10.
What is electrophoresis?
Electrophoresis is a technique used to separate biological molecules, such as nucleic acids, carbohydrates, and amino acids, based on their movement due to the influence 532 of a direct electric current in a buffered solution. Positively charged molecules move toward the negative electrode, while negatively charged molecules move toward the positive electrode.
What wood is the favorite for butcher's blocks?
Because of its resilience, the preferred wood for making butcher's blocks is derived from the American sycamore. Platanus occidentalis, also known as the American plane tree, buttonball, buttonwood, and water beech. The wood of Platanus occidentalis is also used as for veneers for decorative surfaces, fence posts, and fuel.
What names are used for groups of birds?
A group of birds in general is called a congregation, flight, flock, volée, or volley.
How are organelles partitioned during mitosis?
Following the telophase of mitosis, cytokinesis, physical separation of the daughter cells occurs. Although the exact mechanisms are not clear, it appears that larger organelles, such as the endoplasmic reticulum and Golgi complex, undergo fragmentation into small vesicles during mitosis and later reassemble in daughter cells.
What are the major differences between cell division in plants and cell division in animals?
The major differences in plant and animal cell division are in the assembly of the spindle apparatus. The site of spindle apparatus assembly is the centrosome. In animal cells, a pair of centrioles is at the center of the centrosome. In contrast, most plants lack centrioles, but they do have a centrosome. In animal cells, a cleavage furrow forms during cytokinesis, which deepens and then pinches the parent cells in two. Plant cells, which have cell walls, do not have a cleavage furrow. Instead, a cell plate is produced in the middle of the parent cell, which grows toward the perimeter of the cell until it reaches the plasma membrane, dividing the cell in two. A new cell wall then forms from the cell plate.
Why do cells die?
Cells die for a variety of reasons, many of which are not deliberate. For example, cells can starve to death, asphyxiate, or die from trauma. Cells that sustain some sort of damage, such as DNA alteration or viral infection, frequently undergo programmed cell death. This process eliminates cells with a potentially lethal mutation or limits the spread of the virus. Programmed cell death can also be a normal part of embryonic development. Frogs undergo cell death that results in the elimination of tissues, allowing a tadpole to morph into an adult frog.
Where does respiration take place?
Different types of animals have different respiratory organs for gas exchange. For 286 types of respiratory organs are 1. Skin, 2. Gills, 3. Tracheae, and 4. Lungs. Many invertebrates and some vertebrate animals, including amphibians, breathe through their skin. Many of the animals that breathe through their skin, a process known as cutaneous respiration, are small, long, and flattened, for example, earthworms and flatworms. All animals that rely on their skin for respiration live in moist, damp places in order to keep their body surfaces moist. Capillaries. Small blood vessels, bring blood rich in carbon dioxide and deficient in oxygen to the skin's surface, where gaseous exchange takes place via diffusion. Gills may be external extensions of the body surface, such as those found in aquatic insect larvae and some aquatic amphibians. Diffusion of oxygen occurs across the gill surface into capillaries, while carbon dioxide diffuses out of the capillaries into the environment. Fish and some other marine animals have internal gills. Water enters the animals through the mouth, then flows over the gills in a steady stream and out through gill slits. Although some animals with gills spend part of the time on land, they all must spend some time in moist, wet environments for the gills to function. Lungs are internal structures found in most terrestrial animals where gas exchange occurs. The lungs are lined with moist epithelium to avoid their becoming desiccated. Some animals, including lungfish, amphibians, reptiles, birds, and mammals, have special muscles to help move air in and out of the lungs. Some animals have lungs connected to the outside surface with special openings, and do not require special muscles to move air in and out of the lungs. Insects have a system of internal tubes, called tracheae, that lead from the outside world to internal regions of the body via spiracles. Gaseous exchange takes place in the tracheae. Some insects rely on muscles to pump the air in and out of the tracheae, while in others the process is a passive exchange of gases. Some spiders have book lungs in addition to tracheae. Book lungs are hollow, leaf-like structures through which the blood flows. These lungs hang in an open space that is connected to a tube. The other side of the tube is in open contact with the air.
Why do birds fly in formation?
Some birds, like geese and cranes, often fly in V formation because of the phenomenon of wingtip vortices. The lead bird in the V formation breaks up the wall of air that the flock flies into. The swirling air then helps push along the birds behind. However, being the lead bird is hard work, and the leader may then drop back to allow another bird to take on the work of the lead bird. The V formation also gives geese the ability to watch each other and communicate, via honking, about likely landing spots.
What is a redox reaction?
A redox reaction is actually a series of chemical reactions involving the transfer of electrons from one atom or molecule to another. By transferring electrons, the cell is also transferring energy. The molecule that loses an electron is said to be oxidized, resulting in its charge becoming positive. The molecule that gains an electron is said to be reduced, and its charge becomes negative. Electrons do not travel alone in cells, rather, they are accompanied by a proton, H+. A proton and an electron together make a hydrogen atom. Ultimately, a redox reaction involves the loss of hydrogen atoms from one molecule and the gain of hydrogen atoms by another molecule.
In addition, rainforests. When was the symbol of Smokey Bear first used to encourage forest fire prevention?
The origin of Smokey Bear can be traced to World War II, when the U.S. Forest Service, concerned about maintaining a steady lumber supply for the war effort, wished to educate the public about the dangers of forest fires. They sought volunteer advertising support from the War Advertising Council. And on August 9, 1944, Albert Steele, a noted illustrator of animals, created Smokey Bear. Since 1944, Smokey Bear has been a national symbol of forest fire prevention not only in America, but also in Canada and Mexico, where he is known as Simon in both countries. This public service advertising, PSA, campaign is the longest-running PSA campaign in U.S. history. In 1947, a Los Angeles advertising agency coined the slogan "Only you can prevent forest fires." On April 23, 2001, after more than 50 years, the famous ad slogan was revised to "Only you can prevent wildfires." In response to the wildfire outbreaks during 2000, the campaign gained a living mascot in 1950 when a firefighting crew rescued a male bear cub from a forest fire in the Capitol Mountains of New Mexico. sent to the National Zoo in Washington, D.C., to become Smokey Bear. The animal was a living symbol of forest fire protection until his death in 1976. His remains are buried at the Smokey Bear State Historical Park in Capitan, New Mexico.
Are the guard cells the same in all plants?
The guard cells in all plants have the same function. They regulate the exchange of gases and water by opening and closing the stomatal pore. However, structurally they are different. Guard cells in dicots are kidney-shaped, while those in monocots are shaped like dumbbells.
What period of time has come to be known as the Golden Age of microbiology?
The era known as the Golden Age of microbiology began in 1857 with the work of Louis Pasteur. 1822 to 1895, and lasted about 60 years. During this period of time, there were many important scientific discoveries. Joseph Lister's, 1827 to 1912, practice of treating surgical wounds with a phenol solution led to the advent of aseptic surgery. The advancements Paul Ehrlich, 1854 to 1915, made to the theory of immunity synthesized the magic bullet, an arsenic compound that proved effective in treating syphilis in humans. In 1884 Elie Metchnikoff, 1845 to 1916, an associate of Pasteur, published a report on phagocytosis. The report explained the defensive process in which the body's white blood cells engulf and destroy microorganisms. In 1897 Masaki Ogata reported that rat fleas transmitted bubonic plague, ending the centuries-old mystery of how plague was transmitted. The following year, Kiyoshi Shiga, 1871 to 1957, isolated the bacterium responsible for bacterial dysentery. This organism was eventually named Shigella dysenteriae. During the Golden Age of microbiology, researchers identified the specific microorganism responsible for numerous infectious diseases. The following chart identifies many of these diseases, their infectious agent, who discovered them, and the year they were discovered.
What differences are there between cellular slime molds and plasmodial slime molds?
Although both are called slime molds, cellular and plasmodial slime molds have few features in common and differ in their life cycles. Cellular slime molds are similar to amoebas. They move, feed, and reproduce as a single cell. Plasmodial slime molds, Plasmodium, consist of a multinucleate mass of cytoplasm lacking cell walls.
Can human cells grow outside the body?
Human cells can be cultured in vitro, a Latin term meaning outside the body. If the cells are given oxygen and appropriate nutrients. However, these cells usually pass through a limited number of cell divisions. About 50, before they begin to die.
Why can fossils be misleading?
The fossil record is biased toward organisms who were very common, who may have had hard shells or bony structures, and whose species lasted a long time. Therefore, it does not give us a full picture of what species may have been active in the evolutionary past. Fossils are also unlikely to record soft structure changes such as an increase in muscle mass or the development of new organ systems.
How does thin layer chromatography differ from other chromatographic techniques?
Thin layer chromatography does not use a column. The stationary phase is thinly coated on a substrate, most frequently a glass plate. A binding agent such as calcium sulfate is electrophoresis has become an important tool in DNA identification, genetic fingerprinting. Usually added to make the adsorbent adhere to the substrate. A sample is placed on the plate and the mobile phase migrates through. The stationary phase by capillary action, similar to paper chromatography. After separation, the spots may be scraped from the glass for detailed analysis. Or they can be subjected to further chromatographic study.
What are the main types of receptors?
Receptor cells are cells that receive stimuli. Each type of receptor responds to a particular stimulus. The 5 main types of receptors are pain receptors, thermoreceptors, mechanoreceptors, chemoreceptors, and electromagnetic receptors. Pain receptors are probably found in all animals. However, it is difficult to understand non-human perception of pain. Pain often indicates danger, and the animal or individual retreats to safety. Thermoreceptors in the skin are sensitive to changes in temperature. Thermoreceptors in the brain monitor the temperature of the blood to maintain proper body temperature. Mechanoreceptors are sensitive to touch and pressure, sound waves, and gravity. The sense of hearing relies on mechanoreceptors. Chemoreceptors are responsible for taste and smell. Electromagnetic receptors are sensitive to energy of various wavelengths including electricity, magnetism, and light. The most common types of electromagnetic receptors are photoreceptors that detect light and control vision.
How is the gender of alligator embryos determined?
The gender of an alligator is determined by the temperature at which the eggs are incubated. High temperatures of 90 degrees to 93 degrees Fahrenheit, 32 degrees to 34 degrees Celsius, result in males. Low temperatures of 82 degrees to 86 degrees Fahrenheit, 28 degrees to 30 degrees Celsius, yield females. This determination takes place during the second and third week of the 2-month incubation. Further temperature fluctuations before or after this time do not alter the gender of the young. The heat from the decaying matter on top of the nest incubates the eggs.
What tests can be used to determine the presence of lipids?
A very simple test to determine the presence of lipids is the grease spot test. Place a drop of solution on an unglazed, clean sheet of brown paper. Once the liquid evaporates, a spot containing lipids will remain visible. The Sudan 4 test also tests for lipids. It is based on the ability of lipids to selectively absorb pigments in fat-soluble dyes.
How do antibiotics destroy an infection?
Antibiotics function by weakening the cell wall or interfering with the protein synthesis or RNA synthesis of the bacterial cell. For example, penicillin weakens the cell wall to the point that the internal pressure causes the cell to swell and eventually burst. Certain antibiotics are more effective against gram-negative bacteria, while others are more effective against gram-positive bacteria.
What are some beneficial uses of the sclerotia of the fungus Claviceps purpurea?
The sclerotia of Claviceps purpurea are known as the plant disease ergot. Ergot is used pharmaceutically to produce drugs used to induce labor in pregnant women and to control bleeding after childbirth. Ergotamine, an ergot alkaloid, is used to treat migraine headaches.
Who was Linus Pauling?
Linus Carl Pauling, 1901 to 1994, the only person to win 2 unshared Nobel Prizes. 1954 Prize in Chemistry, 1962 Peace Prize, revolutionized the study of chemistry. Aided the development of molecular biology, and made important advances in medical research. He was responsible for determining the molecular basis of sickle cell anemia. Was one of the scientists engaged in the race to determine the molecular structure of DNA. And became a strident advocate for the use of large doses of vitamin C to prevent illness.
What is unusual about the teeth of sharks?
Sharks were among the first vertebrates to develop teeth. The teeth are not set into the jaw but rather sit atop it. They are not firmly anchored and are easily lost. The teeth are arranged in 6 to 20 rows, with the ones in front doing the biting and cutting. Behind these teeth, others grow. When a tooth breaks or is worn down, a replacement moves forward. One shark may eventually develop and use more than 20,000 teeth in a lifetime.
What is meant by genetic determinism?
Genetic determinism is a theory proposing that behavior and character are entirely shaped by genes. Since only genetic material passes from one generation to the next, it seemed logical at one time to assume that all of our traits, physical, behavioral, psychological, were the result of gene sequences. More recent and extensive research has demonstrated the importance of environmental factors in determining how or which genes are actually involved in the development of the individual.
What are the different shapes and functions of the epithelium?
Epithelial tissue consists of densely packed cells. Epithelial tissues are either simple or stratified based on the number of cell layers. Simple epithelium has one layer of cells, while stratified epithelium has multiple layers. Epithelial tissue may have squamous, cuboidal, or columnar-shaped cells. Squamous cells are flat, square cells. Cuboidal cells form a tube. Columnar cells are stacked, forming a column taller than they are wide. There are two surfaces to epithelial tissue. One side is firmly attached to the underlying structure, while the other forms the lining. The epithelium forms a barrier, allowing the passage of certain substances while impeding the passage of other substances.
When was the first plant patent issued?
Henry F. Bosenberg, a landscape gardener, received U.S. plant patent number 1 on August 18, 1931. For a climbing or trailing rose.
Who first developed the modern technique for the isolation of cell parts?
Robert R. Bensley, 1867–1956, and Norman Lewis Hoare, 1902–1958, disrupted the liver cells in a guinea pig and isolated mitochondria in 1934. Between 1938 and 1946, Albert Claude, 1899–1983, continued the work of Bensley and Hoare and isolated two fractions, a heavier fraction consisting of mitochondria and another fraction of lighter submicroscopic granules, which he called microsomes. Further developments led to the development of centrifugal techniques of cell fractionation commonly used now. The development of this procedure was one of the earliest examples of differential centrifugation. It initiated the era of modern experimental cell biology.
What factors contribute to the spread of emerging infectious diseases?
Some of the factors contributing to the spread of emerging infectious diseases include environmental changes, unwarranted use and abuse of antibiotics, and the prevalence of modern transportation, which allows diseases to spread in wide geographic areas very rapidly.
What disease is caused by the protist Entamoeba histolytica?
Entamoeba histolytica, a parasitic amoeba, causes amoebic dysentery, an intestinal disorder. Estimates indicate that up to 10 million individuals in the United States have parasitic amoebas, but only 2 million exhibit symptoms of the disease. In tropical areas, up to half the population may be infected.
What is Gattaca?
The 1997 film Gattaca was one of the first movies to examine the idea that one's station in life is dependent on the perfection of one's genes. The movie explores genetic discrimination and previews the bioethics of using genetic information as a means of identification.
What is the Kelvin temperature scale?
Temperature is the level of heat in a gas, liquid, or solid. The freezing and boiling points of water are used as standard reference levels in both the metric centigrade and the English system, Fahrenheit. In the metric system, the difference between freezing and boiling is divided into 100 equal intervals called degree centigrade, degree C. In the English system, the intervals are divided into 180 units, with one unit called degree Fahrenheit, degree F. But temperature can be measured from absolute zero, no heat, no motion. This principle defines thermodynamic temperature and establishes a method to measure it upward. This scale of temperature is called the Kelvin temperature scale, after its inventor, William Thomson, Lord Kelvin. 1824 to 1907, who devised it in 1848. The Kelvin, symbol K, has the same magnitude as the degree centigrade. The difference between freezing and boiling water is 100 degrees, but the two temperatures differ by 273.15 degrees. Absolute zero, which is minus 273.15 degrees Celsius on the centigrade scale.
Which fungus may have played a role in the Salem witch trials?
The Salem witch hunts of 1692 may have initially been caused by an infestation of a microbiological poison. The fungus Claviceps purpurea, commonly known as rice mud, produces the poison ergot. When ingested, this poison produces symptoms similar to the ones that presented in the girls who accused others of being witches in Salem. Historians and biologists have reviewed environmental conditions in New England from 1692. 1692 and have found that conditions were perfect for an occurrence of rice mud overgrowth. The weather conditions during those years were particularly wet and cool. Ryegrass had replaced wheat as the principal grain because wheat had become seriously infected with wheat rust during long periods of cold and damp weather. The symptoms of ergot poisoning include convulsions, pinching or biting sensations, stomach ailments, as well as temporary blindness, deafness, and muteness.
What are some of the most common enzyme deficiencies?
Lactose intolerance, a condition that results from the inability to digest the sugar present in milk. Lactose is one of the most common enzyme deficiencies. While nearly everyone is born with the ability to produce lactase, the enzyme responsible for lactose breakdown, many people lose this ability with age. Lactase production in adults varies by ethnic group. Glucose 6-phosphate dehydrogenase deficiency is a more serious enzyme deficiency that is linked to the bursting of red blood cells, hemolysis. This deficiency is found in more than 200 million people, mainly Mediterranean, West African, Middle Eastern, and Southeast Asian populations.
What is the latest genome to be made public?
In January 2004, the first draft of the honeybee genome was made available.
What plants are erroneously called mosses?
Not all plants called mosses are bryophytes. Irish moss, Chondrus crispus, and related species are actually red algae. Iceland moss, Cetraria islandica, and reindeer moss, Cladonia rangiferina, are lichens. Club mosses, genus Lycopodium, are seedless, vascular plants, and Spanish moss. Tillandsia usneoides, is a flowering plant in the pineapple family.
What is meant by the term true breeding?
Individuals, who, when bred to others of the same genotype, produce only offspring of that genotype, are called true breeding. In other words, homozygous individuals, AA, are true breeding when bred among themselves, while heterozygotes, AA, are not.
Why is Mendel's work disputed?
There are some scientists who feel that Mendel's work is too perfect to be accurate. Even though the validity of his conclusions are not in doubt. A closer look at Mendel's work suggests that he may not have reported the inheritance of traits that did not show independent assortment, and thus he may have skewed some of his data. A British statistician and population geneticist, R.A. Fisher, 1890–1962, pointed out in 1936 that Mendel's data fit the expected ratios much closer than chance alone would indicate. However, Mendel's published data is comparable to the work of others in his field, and his conclusions are still accepted as part of the core of genetics.
What is the meaning of the phrase ontogeny recapitulates phylogeny?
Ontogeny is the course of development of an organism from fertilized egg to adult. Phylogeny is the evolutionary history of a group of organisms. The phrase ontogeny recapitulates phylogeny originated with Ernst Haeckel, 1834–1919. It means that as an embryo of an advanced organism grows, it will pass through stages that look very much like the adult phase of less advanced organisms. For example, at one point each human embryo has gills and resembles a tadpole. Although further research demonstrated that early-stage embryos are not representative of our evolutionary ancestors, Haeckel's general concept that the developmental process reveals some clues about evolutionary history is certainly true. Animals with recent common ancestors tend to share more similarity during development than those that do not. A dog embryo and a pig embryo will look more alike through most stages of development than a dog embryo and a salamander embryo, for example.
Why is Rosalind Franklin called the Dark Lady of DNA?
Rosalind Franklin, 1920–1958, was a chemist by training. She worked at King's College, London, in 1951 in the lab of John Randall, 1905–1984. Both Franklin and Randall were working on the structure of DNA using the relatively new field of X-ray crystallography. Through meticulous research on the DNA molecule, Franklin took photographs that indicated a helical structure. Randall presented Franklin's work at a seminar where it was then, without Franklin's knowledge, provided to the competitors, Watson and Crick, at Cambridge University. This research was crucial to the detailed description of DNA that was published in 1953. Because the Nobel Prize is only awarded to the living, Franklin, who died of cancer in 1958, did not share the award when it was given to Watson, Crick, and Wilkins in 1962.
Who disproved Lamarck's theory?
In the 1880s, the German biologist August Weismann, 1834 to 1914, formulated the germplasm theory of inheritance. Weismann reasoned that reproductive cells, germ cells, were separate from the functional body cells, soma or somatic cells. Therefore, changes to the soma would not affect the germplasm and would not be passed on to the offspring. In order to prove that the disuse or loss of somatic structures would not affect the subsequent offspring, Weismann removed the tails of mice and then allowed them to breed. After 20 generations of this experimental protocol, he found that mice still grew tails of the same length as those who had never been manipulated. This not only disproved Lamarck's theory of use and disuse, it also increased understanding of the new field of genetics.
What are two of the most common forms of dementia?
The term dementia describes a group of symptoms that are caused by changes in brain function. The two most common forms of dementia in older people are Alzheimer's disease and multi-infarct dementia, sometimes called vascular dementia. These types of dementia are irreversible, which means they cannot be cured. In Alzheimer's disease, nerve cell changes in certain parts of the brain result in the death of a large number of cells. The symptoms of Alzheimer's disease range from mild forgetfulness to serious impairments in thinking, judgment, and the ability to perform daily activities. In multi-infarct dementia, a series of small strokes or changes in the brain's blood supply may result in the death of brain tissue. The location in the brain where the small strokes occur determines the seriousness of the problem and the symptoms that arise. Symptoms that begin suddenly may be a sign of this kind of dementia. People with multi-infarct dementia are likely to show signs of improvement or remain stable for long periods of time, then quickly develop new symptoms if more strokes occur. In many people with multi-infarct dementia, high blood pressure is to blame.
What is a trophic level?
A trophic level represents a step in the dynamics of energy flow through an ecosystem. The first trophic level is made up of the producers, those within the ecosystem that harvest energy from an outside source like the sun or deep-sea thermal vents, and stabilize or fix it so that it remains in the system. The second level would comprise those who consume the producers, also known as the primary consumers. The next level would contain the secondary consumers, those who consume the primary consumers, and so on. Because of the limited amount of energy available to each level, these trophic pyramids rarely rise above a third or fourth level of structure. R. Lindemann, 1915 to 1942, was one of the first ecologists to refer to the trophic dynamics of ecosystems, doing so in 1942.
How did scientists decide that DNA was the genetic material for all cellular organisms?
The proof that the material basis for a gene is DNA came from the work of Oswald T. Avery, 1877 to 1955, Colin M. MacLeod, 1909 to 1972, and Macklin McCarty, 1911. In a paper published in 1944, this group of scientists followed the work of Griffith in order to discover what causes non-lethal bacteria to transform to a lethal strain. Using specific enzymes, all parts of the 5 lethal bacteria were degraded, including the sugar-like coat, the proteins, and the RNA. The degradation of these substances by enzymes did not affect the transformation process. Finally, when the lethal bacteria were exposed to DNase, an enzyme that destroys DNA, all transformation activity ceased. The transforming factor was DNA.
What was the first commercial use of genetic engineering?
Commercial recombinant DNA technology was first used to produce human insulin in bacteria. In 1982, genetically engineered insulin was approved by the FDA for use by diabetics. Insulin is normally produced by the pancreas, and for more than 50 years the pancreas of slaughtered animals such as swine or sheep was used as an insulin source. To provide a reliable source of human insulin, researchers harvested the insulin gene from cellular DNA. Researchers made a copy of DNA carrying this insulin gene and spliced it into a bacterium. When the bacterium was cultured, the microbe split from one cell into 2 cells. And both cells got a copy of the insulin gene. Those 2 microbes grew, then divided into 4. Those 4 into 8, the 8 into 16, and so forth. With each cell division, the 2 new cells each had a copy of the gene for human insulin. And because the cells had a copy of the genetic recipe card for insulin, they could make the insulin protein. Using genetic engineering to produce insulin was both cheaper and safer for patients, as some patients were allergic to insulin from other animals.
What is an intron?
An intron is the part of the RNA transcript that is removed by the spliceosome and therefore remains in the nucleus.
How does an animal's energy budget affect its behavior?
Every animal has a finite amount of energy available for use in a unit of time. This energy usage is its metabolic rate, measured in calories or kilocalories, plus the energy required for life activities. The energy budget places limitations on an animal's behavior. For example, a lizard, which is an ectotherm, cold-blooded, uses less energy because it does not maintain a constant body temperature. Ectotherms such as amphibians and reptiles control body temperature by behavior. Endothermic, warm-blooded, animals, e.g., birds, mammals, have a higher energy budget, most of which is used to maintain internal body temperature. Thus, endotherms require more energy than ectotherms of a similar size, and so may spend a greater portion of their day searching for food. Other factors that influence energy requirements are age, sex, size, type of diet, activity level, hormonal balance, and time of day.
How does the mark recapture method estimate a wildlife population?
Researchers set traps to capture a population sampling in a given area. The captured animals are marked, or tagged, and then released. After a certain amount of time the traps are set again. The second time, both marked and unmarked animals will be captured. The proportion of marked to unmarked animals gives an estimate of the size of the entire population. The equation used to estimate the number of individuals in the population is marked individuals x total catch second time n equals recaptured marked individuals, where n is the number of individuals in the population. A disadvantage of this method is that it assumes that a marked individual has the same chance of being trapped as an unmarked individual. Individuals that were trapped once may be wary of traps, or they may seek traps since they have learned that traps provide food.
What is a stem cell?
Stem cells are undifferentiated cells, meaning that they do not have a specific function that are capable, under certain conditions, of producing cells that can become a specific type of tissue. Stem cells present in adult humans are found in bone marrow and other tissues, such as fat. However, most research interest is focused on stem cells present in fetal tissue, which, in a laboratory setting, can reproductively divide indefinitely and be stimulated into becoming a variety of different cell types. The potential benefits of stem cells have made the research of these cells an exciting research topic.
What types of samples can be used for DNA fingerprinting?
Any body fluid or tissue that contains DNA can be used for DNA fingerprinting, including hair follicles, skin, earwax, bone, urine, feces, semen, or blood. In criminal cases, DNA evidence may also be gathered from dandruff, from saliva on cigarette butts, chewing gum, or envelopes, and from skin cells on eyeglasses. What is the largest forensic DNA investigation in U.S.?
What are the advantages of gel electrophoresis?
In gel electrophoresis, the molecules are forced across a span of gel, usually agarose. Gels are easy to modify and have an excellent power of separation. The frictional force of the gel material acts as a molecular sieve, separating the molecules based on their size.
Is there a link between antibiotic use in animal feed and the increase in bacterial infections becoming resistant to antibiotics in humans?
Scientists have discovered a link between agricultural use of antibiotics, particularly in animal feed, and the increase of foodborne infections in humans who consume products derived from these animals. Resistant bacteria present in animals can survive the slaughtering and meat packaging process. Undercooked meat will harbor these bacteria and when eaten can cause illness in humans. To further complicate the situation, the antibiotics used to treat humans made ill by infections may be similar to those used routinely in animals, rendering them less effective.
What are meristems?
Meristems, from the Greek term meristos, meaning divided, are unspecialized cells that divide and generate new cells and tissues. Apical meristems, found at the tips of all roots and stems, are responsible for a plant's primary growth. The vascular cambium and cork cambium are the meristems responsible for a plant's secondary growth.